Okay, welcome everyone to the Genomic AI Network and Alan Turing Institute webinar series for AI in genomics. Today's topic is going to be the regulation of AI devices in genomics, and that's a really interesting tightrope between innovation and patient safety. And here to talk about that topic today, we're delighted to be joined by two of the finest from Hardian Health, which is a healthcare consultancy company based in the UK, Doctors Ankeet Tanna and Felicity, and I'll let them introduce themselves. For the audience, if you have any questions, please put them in the Q&A section at the top, not in the chat, so that they can be answered and upvoted during the presentation itself. Thank you very much, and I'll hand over to the team from Hardian Health.
Thanks, Alex. Just before we start, is there an opportunity for attendees to raise their hand and ask questions directly, or would you prefer just on the Q&A?
I think Q&A makes more sense, and I'll collate them at the end for when we do that.
Great, thanks. Well, in that case, it's great to be here. My name's Ankeet. I'm a doctor by background with previous experience developing machine learning tools for neurodegenerative diseases. And I'm a senior consultant at Hardian Health, where I lead the clinical team and helping our clients in determining the regulatory strategy and clinical evidence they need to get onto market. And I'll hand over to Flick to introduce herself, and then we'll move on to actually doing the presentation.
Yes, hello everyone, and thank you for inviting us to be here today. I'm Felicity. I'm also a doctor by background and integrated in biomedical engineering, but I've been with Hardian for the last 2.5 years in the clinical team, working alongside Ankeet and the rest of our multidisciplinary team. So it's really lovely to be here today, and thanks for the invite.
Great. So before we go on, just a little bit about ourselves. So we're a multidisciplinary consultancy. Flick and I are from the clinical team, but we have technical and regulatory team members, ex-software engineers, quality assurance managers, a whole market strategy team, health economics, and intellectual property. And our whole aim is to basically be an end-to-end service for people who are building novel technologies and bringing them into the market. So we've worked in a lot of different spaces. Next slide. Including AI genomics. Radiology AI is obviously a big space for software medical devices and digital therapeutics as well. So today we're going to be talking about an overview of the regulations. Particularly, I'll be covering what the regulations are meant to do and how to qualify and classify medical devices. And then Flick will continue to talk about how to conduct clinical evaluation of medical devices. And we'll briefly touch on the documentation requirements and take any questions as we go along. So if there are any relevant questions that come into the Q&A, we'll try and answer them as we go along. But if they require a bit more explanation, we might leave them to the end. So without further ado, we'll talk very briefly about why regulators regulate. This is important to start with because often people see regulation as a tick box exercise and not necessarily understand where the regulators are coming from and what they're looking out for. And so when we think about what regulators are looking for, I like to split it into 4 main buckets. So the first and probably most important bucket is risk and safety. So doing no harm essentially is the most important thing that regulators want to see. Obviously, with great clinical benefit, there may come a bit of risk, and that's okay. Regulators are happy to see that, but they want to essentially see that you've mitigated risks as much as possible in the context of the benefits that you're trying to provide to patients. The next bucket is quality and consistency. So are you able to deliver the same results or deliver the same product every time and in a consistent way? And are you able to make changes in a safe way to make sure that the service you deliver or the output of your any devices that you have meet the standards that are required for clinical practice? And then the third bucket is evidence of effectiveness. So as medical device manufacturers, they will have to make claims about what they do, and regulators are there to make sure that they have the evidence to substantiate and back up those claims. And the fourth bucket is hopefully one that we don't have to speak about too much, but if you don't do the things in the first 3 buckets, the regulators have the power to remove bad actors from the market, whether that's intentional bad actors or unintentional bad actors. So those are the 4 key buckets that we're looking at. And essentially the whole process of getting regulatory clearance for AI devices or any type of device really is to try and answer those sort of, those buckets of questions that the regulators will have. So now that we've done that, we can move on to defining what a medical device actually is. So a medical device is basically apparatus, software, reagents, machines, instruments, basically any noun that is not a drug and that has a medical purpose. So that can range from syringes and even the trolleys that dialysis machines are put on, all the way to very complex genomic AI pipelines. And it all falls under the same regulatory framework of the medical device regulations. Then when we think about what software as a medical device is, the way to consider it is basically software will always have some sort of input data. It will perform some sort of action on it. And as long as that action or that process is more than just simply searching that data or just presenting that data or storing that data, then you probably have a medical device functionality, as long as the output, next slide, is related to keywords like diagnosis, monitoring, treatment, alleviation, or related words. And importantly, it has to be for the benefit of for the benefit of individual patients. So as long as you have a high functionality piece of software, doesn't need to be AI, it can be sort of classical machine learning or even something that's just not easily verifiable by hand, and it has a medical purpose. If you tick those 2 boxes, then you're a medical device. Now, what's interesting is actually in the genomic AI space, a lot of the things that you may consider building or buying in your service are probably in vitro diagnostic devices. So an in vitro diagnostic device is essentially a device which analyzes a sample derived from human tissue. And the regulations basically say if you have software that also interprets the output of another in vitro diagnostic, that software is also going to be classed as an in vitro diagnostic. And some of the regulatory nuances are different, which is why it's important to make this point. Just because you're building software, because you're building it in the genomic AI space, it's more likely to be an in vitro diagnostic because it's taking in input data from another in vitro diagnostic device, for example, a sequencing machine. So this is an example from the medical device software guidance where basically there is this informatics pipeline. MDSW stands for medical device software, that takes NGS data and interprets the number of alterations or rearrangements and outputs things like tumor burden. And essentially what the guidance basically says is, even though this is a medical device software, because it's substantially driven by the outputs of a next generation sequencing machine, it counts as an in vitro diagnostic medical device. So you can't see the steps here, but we'd be happy to share the guidance which sort of gives this example. If we move on to the next slide. So everything that we've talked about so far is what we call qualification of a medical device. So do you actually qualify as a medical device or an in vitro diagnostic? And once you've determined that you qualify as a medical device or an in vitro diagnostic, the next thing to do is classify it. And the way the regulators do this is by risk. So in medical devices, the risk classes are 1, 2, and 3. Class 1 devices are low risk, 2 are moderate risk, and 3 are high risk. And the important thing to remember is essentially the higher the risk, the more evidence of effectiveness that you need to show, and evidence of safety, of course. Now, the classification for in vitro diagnostics is a little bit different. In the EU, it's not 1, 2, 3, it's actually class A, B, C, and D. And the reason I'm mentioning the EU regulations is because currently the UK regulations are different, but we anticipate that in about 8 months' time, in June 2026, the UK is going to align with what the EU classification criteria are for in vitro diagnostics. So generally, the way the classification rules work is at the end of the regulations, there's a whole bunch of rules that you have to follow and say, okay, well, does rule 1 apply to what you're building? Yes, no. Does rule 2 apply? Yes, no. And essentially, the rules are structured not around the complexity of a device, but more around the clinical risk or the clinical space. So devices used for testing for blood typing would be a higher risk than devices that are used for measuring simple analytes in a blood sample. In the case of genomic AI, genomic testing and associated software would generally fall under Class C, which is the third highest bucket of classification. And so if we move on to the next slide, important examples might be companion diagnostics. So these are tools that you might use to determine whether, often cancer therapies are suitable to be prescribed to patients. So here are some specific examples from the guidance document again. And the next slide. Genetic testing in general. So anything from chromosomal conditions or predicting genetic risk, or pharmacogenomic testing, which is becoming an increasingly popular space in the genomics area for AI. All of these would count as Class C medical devices, and any AI associated with these kinds of tests would also therefore be Class C, because essentially if you are using software that influences the use of a Class C medical device, that software will also be Class C. So the important thing that I wanted to highlight here is it's much less about AI and machine learning or the complexity of the software, but it's much more about the intended purpose, which Flick will talk about a little bit more. What you're using the device for will determine its risk classification a lot more than how it works. If we move on to the next slide, I just wanted to give a couple of examples of technologies that already have Class C in vitro diagnostic certification. So some of you may have come across these or may not have, but the Seq1 platform essentially is a next generation sequencing and data analysis platform that received Class C IVD certification about a year ago. The next slide, variant interpretation. QIAGEN also successfully got a CE mark for a Class C IVD for their platform, which helps clinicians and healthcare professionals with variant interpretation, so reading VCF files. And that's just an example of kind of how these companies have gone about. And it is, you do see these products on the market, which are very software driven, but still follow the in vitro diagnostic regulations. So I will leave it at that. So I've talked about qualification and classification. And then the next steps are actually how to evaluate the safety and effectiveness of these tools. And I'll hand over to Flick to talk about that.
Great, thank you, Ankeet. So yeah, as Ankeet's just covered, we've obviously gone through the qualification and the classification, and now we're going to go through a bit of an overview of the clinical evaluation process, which is actually known as performance evaluation for in vitro diagnostics that were just mentioned previously. So the overall goal with this process is to write a clear narrative that's written across multiple chapters. So regardless of the tool or the product that you're developing, these chapter headings will always be roughly the same, but actually the content in each chapter will be different depending on the tool that you're developing. So over the next sort of 15-20 minutes, we're going to take a deep dive into each of these chapters. So this is not really a regulatory chapter, I would say, but it's a very important step to start with. So the key thing is to think about, why are you building this product in the first place? What is the clinical unmet need that you're trying to help solve in the healthcare system? So the interface, I would say, with the regulatory process is actually that if you have a clear unmet need that you're developing a product for, this can actually help you more clearly define the product that you're wanting to build, and help you define exactly like how you're going to fit that issue that you've identified. The other concept to think about, obviously right at the start, is whether you have a commercially viable value proposition. So are you building a product that will ultimately be reimbursed and adopted in the healthcare setting that you're targeting? Because we've obviously seen in other areas, such as in the radiology AI space that's been mentioned earlier, we've had a lot of sort of swelling of that market. We've had a lot of regulatory approvals in that market over the last 10 years, but actually we're seeing a slow clinical adoption and slow reimbursement for this space. So it's really, really important to start with your why. So, okay, so you've identified your clear unmet need, you've decided to develop a product that has a clear value proposition, and you've decided it's worth now developing into a commercial product. So this is where your regulatory clinical evaluation process comes in. And the first step in this is conceptualization. So this all comes back to one of the key documents in a regulatory medical device file, which is called the intended use. So the purpose of this step is to define exactly what your product does. So who the users are, which patient population you're targeting, the severity of the disease that you're targeting, so that a clinician, say it's for a clinician to use, they know exactly which kind of medical indication that they would use it in, which clinical situation they would use it in, and whether or not it would be suitable for the patient in front of them. And it's not just, it's best to not think about it, it's not just a single sentence or a single paragraph, but it's a complete foundational document for your medical device file that the rest of your clinical evaluation process then stems from. So for example, we've got these headings on the screen, which are some of the key headings in an intended use. And the patient population, for example, it wouldn't be enough to just say that it could be used in adults. You'd have to be a lot more specific than that. So what age range would it be suitable for? Are there any contraindications that it couldn't be used in? And the key thing to note is that anything that's described in this intended use is then the evidence, that's the foundation of the evidence that you then need to develop in later steps throughout your process. So it's important to be extremely clear at this step. So once you've sort of defined your intended use, the other aspect of that is thinking about the clinical benefits that your device will impart on the individual patients. So normally for medical devices, this concept of clinical benefit is intended to be a direct clinical benefit to the patient. So should it somehow improve outcomes for that patient. However, it has been acknowledged in guidance from the EU regulators that actually software devices often may not provide a kind of direct benefit to a patient, but actually the provision of accurate medical information by the software then allows a benefit to be imparted through, say, further diagnostic options or treatments that are then made available as a result of that accurate medical information. So for often for software, especially for kind of IVD software that's producing a kind of like a specific accurate measurement or accurate output about a gene or something like that, that would then be an indirect benefit because then a patient's then got access to a certain ongoing diagnostic or final diagnostic option. So to give an example, maybe a developer's making a genomic AI tool that looks at a patient's genetic predisposition to a certain cancer. The developers may want to claim that actually they can just provide accurate information about that person's genetic predisposition, but actually the patient benefit is then provided by, say, a diagnostic option or the ability to undertake preventative treatment for that patient. So that has a positive impact then on that patient. So that's the kind of nuance to think about. The other thing to note on the clinical benefit side is about this. Often it's a stumbling block that we come across is the difference between features and benefits. So it's important to think about, your benefits are a benefit to the patient, but they're provided through the features of your product. So that's one way to think about it. Right, so we've now looked at the why, so we've mapped out the what in conceptualization with the intended use, and the next step is to do the how. And so in regulatory terms, this is done in 3 key steps, which form the pillars of the clinical evaluation process or performance evaluation process. So the first step is scientific validity. So this is usually done through a regulatory compliance systematic literature review, where what you're trying to do is demonstrate that your kind of software output is associated with a target clinical condition or a physiological state, and that association is well-founded in science or a clinically acceptable association. So the way that you sort of go about that is by, 1, undertaking a systematic literature review of academic literature. Number 2, performing a competitor analysis. So you're looking at any on-market devices that are similar to yours or doing a similar, performing a similar process or have similar outputs. And you're then looking at adverse events for those on-market devices. The other part of it is as well looking at the state of the art. So how is this problem currently solved in the healthcare setting? And then using all of that information to determine what the performance criteria need to be for your device based on all of this, this found knowledge that you've done in the literature review. If there's insufficient literature available, say it's a very novel area and there's not a lot of literature on that topic, then you can also use things like proof of concept studies, using your device to kind of plug that gap in the scientific validity. So we've done step 2, we've completed a systematic literature review, and we've determined that there's valid clinical association between the device and the target clinical condition. The next step is to build the final product. So between steps 2 and 3 is where you need to kind of lock in your final software, your final product that will be on the market and in the hands of clinicians and patients. So once you've done this, you're then ready to move on to the next step, which is called analytical validation. So the main part of analytical validation is about demonstrating and clearly documenting for an external reviewer that each component of your product works as it's intended to do so, and is it technically doing what it's meant to, basically. So for software, this can often be software verification testing, which is, unit testing of each component of your software to make sure that each component is doing exactly what it's supposed to be doing and that there's no kind of issues or bugs that have kind of arisen. This is kind of this all stems again from the intended use. So a lot of this is about developing. What you do is develop a matrix of software requirements, basically based on the product requirements that you've described in your intended use. You're then pulling in requirements about risk mitigation features that have been identified through your risk management processes, through the systematic literature searches, the adverse event searches, and you're then systematically checking that each component passes all the tests and everything's performing as expected. So we obviously work mostly with software devices, but we do also often work with hardware devices as well. And the important thing to note is that there's also, hardware testing that needs to be done at this stage. So some IVD products in the genomic AI space might have hardware components such as, self-test devices that go direct to the patient or, used with a clinician. And there are various ISO standards that are relevant for hardware testing. So particularly things like biocompatibility. So if there's any direct contact with a human, so direct contact with the skin, if it's a wearable or something like that. And then, things like compliance with lab standards. So there's various lab standards that are developed by ISO. So ISO 15189 is one example, which is the quality and competence for medical laboratories, which I'm sure you're all aware of. So it's important to note at this stage is that it's the manufacturer's responsibility to determine which ISO standards apply to their specific product in question. So you need to then have a look at what the ISO standards are out there and say which ones apply to my product, and then make sure that you're compliant with all of them. Great. So you've done your analytical validation, you've checked that each component's working as it's intended to. You've got your final product, it's passed all of its internal testing, and this is where we move on to step 4, which is the final step in the clinical evaluation process. And it's often the thing that people think of when they think of clinical evaluation of medical devices. So the question that you're trying to answer is, does your final product deliver the clinical benefits that you've defined at the start as intended on a representative patient population? So in the hands of real world users. So the representative patient population, again, this is linking back to the intended use. So that's the population you've defined in your intended use statement. And then the real world users, the users that you've defined in your intended use statement, which really just it shows how important that intended use statement is at the start. So this is the step where you're conducting your pivotal clinical investigation. So this could be something like a retrospective or prospective clinical investigation, AI external validation, and also summative usability testing. The tricky thing with step 4 is that the exact kind of the activities that you need to take and the scope that you need to go through is highly variable depending on your product and depending on what data you have available to you. So, for example, say you have a tool which is just maybe it's identifying a particular genetic variant. There's loads of literature out there in the systematic literature review, and you've got a very well-founded valid clinical association, your only claims is that you're saying you can accurately determine that variant with an X amount of accuracy. Maybe you could just do a retrospective evaluation, provided you have all the data available to cover your patient population, and just demonstrate that you can accurately identify that variant in question. However, if you're then doing something a lot more novel, say you're recommending a combination of drugs to treat different cancers which are not well described in literature, and you are kind of doing something very novel that's not, well founded, then you would definitely, or very likely, to need a prospective study there to evidence your claims that you're making. For this, obviously, we're focusing on AI, and the important thing for AI tools is that when you're doing an external validation, this has to be on an independent data set. So it can't be data that's been used in the training of the device. And it's also best practice to use kind of multi-center data and it must cover the full breadth of that patient population that we've described earlier. And then the other aspect of step 4 is something that's perhaps less well known, I would say, is summative usability validation. So this involves testing your final product in the hands of, say, 15 to 20 users. So these are the target users that you've described in your IUS. So what you're trying to do is observe them performing key tasks. So you're trying to evaluate the usability of your device in a very, measured and predefined way. So you're trying to demonstrate and write up a report showing that your product meets those predefined usability testing requirements that you've set. And often these requirements are driven by, maybe you've identified some potential human factor errors that could occur with your product, and you've put some risk mitigation measures in place. You're then testing to make sure that those measures work, and that your product meets that usability requirement there. So I've mentioned this a couple of times, but it's so important. So any claims that you make, you have to provide evidence for. When you go to a notified body, who is the body that will review your medical device file, they'll expect to see anything written in the intended use, any claims that you're making, say in marketing materials, on social media, on your website, at conferences and things like that. All of that kind of evidence, they can say, where's your evidence to back up that claim that you're making? So we're just going to touch on briefly clinical investigation design. It's a very common kind of set of questions that we get in sort of day-to-day from clients is, number one, do I need a retrospective or prospective study to evidence my claims, and what kind of sample size do I need? And the difficulty with this is that often the answer is it depends. So the best way to go about this is to just start from first principles and remove kind of any preconceptions or pre-ideas that you've had already built up, and just start from scratch again. And think about what actually is the claim that you want to make. Are you wanting to improve clinical outcomes for patients, or do you just want to claim that you provide accurate medical information? Or maybe you want to claim that a clinician using your device is better than when they're not using your device. So it's really important to just go back to kind of first principles. And a lot of this will also link back to what the unmet need is. So, how are you going to solve the problem that you've identified? And then, what claim do you need to make to be able to solve that problem? And then that can help you think about the exact study design that you need to provide that evidence for that claim. The next question that you can think about is, what data do you already have? So, what's come up in the literature review? Are there any gaps? The important thing to think about, obviously, is if you don't have, sufficient data at present, you may need to limit what your claims are. You may need to limit your intended use, or you need to go out and collect that data to be able to evidence your claims that you want to make. So that's an important thing to think about as well when you're thinking about your investigation design. And then the other thing to think about is the benefits and risks of different study designs. So the actual study design is often driven by the findings from your literature review back in step 2. So, and also maybe what competitors have done, what's regulatory acceptable for that kind of device. And the other thing to consider is that if you're conducting a research on a uncertified medical device in the UK, and often in many other countries it's the same process, you have to obtain what's called a notice of no objection from the MHRA. So this is where you submit some of your documentation, including your protocol and a lot of your risk documentation about your product up to this up to this point, basically up to step 4. And then they will determine whether they feel that this study can go ahead based on the benefits and risks that you've presented to them. And they're able to then give you the green light to go ahead with the study. So they're all important points to think about when you're looking at your investigation design. So between steps 4 and 5 is where you then submit to your notified body or approved body in the UK. And what you do is submit a whole package of documentation to the notified body, and it takes maybe up to 6 months. So what happens is you go through 3 rounds of questions with a notified body. They can come back with questions, they can ask you for clarification, and then that can go on for 3 more rounds, basically for 3 rounds in total. And that whole process can take around 6 months, depending on the complexity of the device. Between 4 and 5, you then hopefully you submit to your notified body, you hopefully get your CE certificate in between 4 and 5, but that's not the end of the process. So once you're on the market, there's still ongoing activities that you have to do from a performance evaluation process perspective while you're in the post-market period when you're on the market. So we're just going to have a look at some of those post-market activities that you may need to conduct. So the idea of post-market is to look at whether your product continues to perform safely in the real world. So is the performance and safety maintained when it's being used in real world conditions with real world users in the messy healthcare system that is surrounding it? So a few of the things that you do, there's kind of 2 sides of post-market activities. One is post-market surveillance. So this could involve gathering user feedback. So you could proactively gather user feedback, or have feedback buttons available in your software so that people can report any issues or bugs that they come across. You have to perform surveillance for adverse events or any bugs that come up. If you see any reports, even say like if you had an app and it was on Google reviews or something like that, or Apple reviews, you'd have to have a look at them and make sure that any issues and things that are coming up, that you're keeping abreast of them and actioning them internally if anything comes up. The other key part about this is complaints. So you need to have, as part of your QMS, internal procedures in place to take corrective actions if any major complaints or issues are identified or reported by end users. So that's your post-market surveillance. That's really important. And then the other aspect in the post-market period is post-market clinical follow-up. So the notified body will expect you to have a plan in place as to whether you need post-market investigations, and if you decide you don't need them, then you need to provide a justification for why not. Often with kind of novel genomic AI tools, you're going to need some kind of post-market clinical follow-up investigation because obviously there's not been many tools in the market yet, and so we don't know exactly what happens in the real world. So the kind of things that you can do in your post-market clinical follow-up is conducting more investigations, looking for, say, drift in performance in certain patient groups, looking for variability between different sites. Say maybe a DGH might have a different kind of performance compared to a tertiary centre that's got a very advanced genomic service. So it's important that all of that information is, aware to you, and you're keeping an eye on everything in the post-market period, so that you can action any kind of risks or issues that come up and make sure that they're not then applied to, they don't then spread to other users and that kind of thing. So we've sort of done a whistle-stop tour there of the defining our value. We've looked at the conceptualization process, where you're writing your intended use and your plan for what you're going to do to generate evidence. We've then looked at these 3 pillars of clinical evaluation, so scientific validity, analytical validity, clinical validity. And then we've looked at what you're doing in the post-market period to monitor that ongoing performance and safety in the real world. So each of these steps that we've gone through link to specific regulatory documentation. So these are just a kind of snapshot of the wider documentation. There's a lot more that goes into it. But for example, we've got our intended use in step 1 and our performance evaluation plan. We've got the literature review that we've mentioned. And then, for example, analytical validity, we've got the software verification plan and software verification reporting. We've also got our user interface evaluation plan and report. The clinical investigation terminology in medical devices is that clinical investigation plan and clinical investigation report. And these are basically the equivalent of, say, a protocol and a report for a clinical trial. And then we've obviously got PMS and PMCF, which we've discussed just now. So what happens is actually all of these documentation then feed into one big narrative story, which is a large document called the Performance Evaluation Report. So this is a standalone document where you get this whole narrative story from start to finish, with information from each of these chapters that we've discussed is pulled into this large narrative report. And ultimately, external reviewers should be able to read that report and understand the whole narrative from start to finish. I just saw PMS and PMCF pop up, but it's post-market surveillance and post-market clinical follow-up. So ultimately, you want to get to your PER, which is your performance evaluation report, and be able to conclude that your product is safe, effective, and cyber secure, and that the benefits of this device outweigh the risks, the potential risks, and that any risks have been mitigated against, and that at this point, it's safe and effective to be on market. And so the important thing as well to note is that while this looks like a line, it's actually more of a circle. So as we've mentioned, the post-market information then feeds back into your kind of clinical evaluation process, and you're continually updating this cycle with new and emerging information. So there might be new literature so you're updating your literature review, annually, and then looking at that new literature, new competitors, new adverse event data, user feedback, complaints, data from your PMCF investigations, and all of this is then a cyclical process which carries on throughout your device lifecycle. So the entire time your product's on the market, and then often some of this is you need to have some of this information continuing after your device comes off the market for any liability or complaints issues. But it's important to think of it as not just a process, but a continual cycle. So that is the kind of overview in a nutshell. I'll just wrap up what we've gone through, and then we'll have time for lots of questions at the end, which I'm sure everyone will have. I've seen a few pop up in the chat already. So just to summarize, technology, if you're building technology that's built for a medical purpose, it's probably a medical device. So for genomic software use cases where we're using tissue samples or blood samples, saliva samples, and that's being used to make decisions or provide an output, that's probably going to be an IVD medical device software, as Ankeet covered in the first half. Really, really important to be clear about your intended use, your features, and your clinical benefits. So these are really key things that will help you in determining what evidence you need to generate, and make your life a lot easier and the process a lot simpler. And like being really clear about what the uses are, what the features are, and what the benefits are, and separating those concepts out quite clearly. And then I've mentioned this, but I think it's such an important point, is to remember that the clinical evaluation is a living, breathing process. It's not just one study that you do. It's not just one and done. It's a constant process throughout the life cycle of your device, and it's a story that you're trying to tell to the regulators, to your customers, that you are, have a culture of clinical evaluation that's ongoing, and you're continually maintaining the safety and performance of your device on market, essentially, for the benefit of patients and clinicians. So I think we're okay to time based on Alexander's timings at the start, but we've got plenty of time for lots of questions, and I've seen a lot pop up, so I don't know if we can field some questions now.
Of course, thank you. First, I hope our audience will join me in a digital round of applause for a very informative talk from colleagues at Hardian Health. And we'll start off with the questions. If you could put all your questions in the Q&A section, please. The Q&A, not the chat section. Mark B from Storgene says, in recent NHS Genomic Medicine procurement draft document, there's a strong emphasis on suppliers having ISO 15189 medical laboratories requirements for quality and competence. While that's appropriate for wet labs, there was no mention of any standards or requirements for digital systems. It's a pity they haven't acknowledged frameworks for bioinformatic tools. I did provide that feedback, but please be aware as a community that this could be a barrier to adoption once a procurement begins. Aside from ISO 27001, are you aware of any standards currently applicable to digital clinical genomic platforms or tools?
Yeah, I'll take that question. Thanks, Mark. Good to see you on the call. Yes. So I think for software IVDs, the standards that would apply would basically be the same standards as software medical devices. So 27001 is correct for information security. And normally what you would try to do is incorporate 27001 into your quality management system, which would need to follow another ISO standard called 13485. There are a few other standards which would be applicable. So I'm going to rattle out some numbers, but I'll share a link as an answer to your question, which has all of them from an insight piece on our website. But risk management for medical devices is ISO 14971. Clinical investigation, design, and conduction is ISO 14155. That would still be applicable. The most important one for digital and bioinformatics tools is a standard called IEC 62304, which basically maps out exactly how you should be documenting your software development lifecycle processes and what kind of testing you need to do. There is likely so those are sort of the main. I'll just post it as a response to your question now. So those are the main standards which are essentially mandatory, and everyone will need to follow if they're building software medical devices. There are additional standards which are not legally required, but may be helpful to point to for how you've conducted your tests, for example. So there are particular standards on how to run testing for things like sensitivity analysis or margins of error, etc. But the list that I've shared in the link is the sort of the core set of standards.
Thank you so much, Ankeet. The next question comes from Sean Palmer. Does the clinical validity stage have to be conducted as research, i.e. working with an academic institution and an NHS organization under research ethics?
Flip can answer that.
Yeah. Sorry, I thought you were going to say something, Ankeet, then. So yeah, the clinical validity step. So it really depends on the investigation that you're conducting. So if you're doing a retrospective study, you may kind of be able to do it internally. It depends kind of on your internal expertise as well in conducting the study. So any investigation that you conduct for medical device safety and performance has to align with ISO 14155. So that's the really key point. If you're involving participants, you still need to comply with all of the standard, good clinical practice, ethics, HRA approvals, etc. that apply. But it really just depends on what internal expertise you have. But the key thing that people often overlook is this need for an MHRA notice of no objection if you're conducting an investigation in the UK. So that's where you're submitting to the MHRA to say, yes, you can have the green light, your device is safe enough and effective enough that we're happy for you to continue to conduct your investigation on real participants in the UK, basically. I hope that answers the question. I don't know if you had anything to add, Ankeet.
Yeah, I would say that the majority of our clients, if they are doing some sort of prospective evaluation, do end up partnering with an academic institution or an NHS trust. If you are doing a retrospective study, that's perhaps less likely, but often it depends on, as Flick said, whether you actually have that data as a company to be able to run that, and obviously have the appropriate permissions to use that data for commercial use. So I would say, yeah, more often than not, there is a partnership with academic institutions, unless the company itself has the resource and the capacity to run those studies independently.
Thank you. Francis Pearl asks, how does Class 2A relate to Class C? So this would be the parallel systems of medical device classification and their appropriate risk.
So,
yeah.
Go ahead. I'll go. So Class 2A, so the risk classifications that Ankeet shared earlier, the Class 1, 2A, 2B, 3, relate to medical devices. So that's slightly different from IVD devices. So the IVD classification is A, B, C, D. So there's actually, more or less roughly a C is like a Class 2, but actually there's not really much comparison between the two. It's just that if your device qualifies as an IVD, you would then follow the Class A to C classification. If it classifies as a medical device, you'd follow class 1 to 3, and it's separate regulations in the EU for medical devices and a separate regulation for IVD devices.
A question from me, as a consultancy firm in this space, what do you think the MHRA should be doing over the coming years with regards to making the guidelines and legislation more legible and easier to follow for companies going through the process of certification?
That's a great question. I think we're not lawyers, but it seems like the law has to be written in a certain way for legal purposes. Obviously, and the language they use is very specific, but often the consequence of that is it can be quite inaccessible and difficult to interpret if you're not sort of in that legal space. The MHRA does do a very good job of providing guidance documents, as does the EU Commission. They provide a lot of guidance documents with a lot of examples to explain how the law is implemented and what they interpret the law as. And those documents are a lot more accessible and easy to understand. And we rely, as a consultancy, we rely on them a lot as well. In terms of what could be done to improve, I think, as you know, it's a bit of a balance because the guidance documents are official, which means that a lot of work goes into them to make sure that they are comprehensive and that they're internally valid. But that does mean it does take a lot of time for them to be updated and new documents and guidance to be released. So if there was anything that the MHRA or the European Commission could do to increase the cadence of updates or increase the level of guidance, then that would be very helpful, I think, for manufacturers to stay up to date on what the current thinking is from regulators. But that just does take some time.
Oh, can I just jump in there as well? I do think that we have seen in recent years, we have seen a slight shift in terms of the guidance that's produced. So earlier this year, there was a digital mental health technology guidance that came out that was, in my opinion, a lot more accessible than some of the previous guidance documents that have gone out. So I think there is a move internally to, make these guidance documents more accessible. And there are obviously, we're in a kind of uncertain period at this point with the potential, the proposed changes to the UK regulations next year, but there's also proposed changes to IVD regulations coming up in the next year or so as well. So I think there is movement and things happening, but I appreciate that can also be confusing when the changes are happening in quite quick succession. But hopefully we're moving towards more transparency.
Thank you. One more from Rebecca Haynes. Hello. This is from the procurement point of view. Sorry to have missed the first part of the presentation, but in procuring software for the use in the genomics lab, what certification and documentation should be requesting from the supplier to be confident that it meets the medical device requirements?
I mean, the ultimate thing you should be asking them for is their certificate of compliance with the medical device or in vitro medical device regulations. So this is a certificate that they will have that would be issued by a notified body. And that certificate basically represents the fact that an external independent party has reviewed all their documentation with regards to risk, safety, effectiveness, and quality. And they agree with the classification of the device as being appropriate. So if you have that, certificate, or they are able to provide that, then that would be one of the most important things to see. And then, depending on exactly what kind of device it is, you may want to ask for additional standards that they comply with. For example, ISO 27001 for information security, and ISO 15189 if they are running samples in their lab on your behalf.
All right. One more minute for the last minute questions coming in. And can I ask, from Hardian's point of view, without revealing anything that's NDA'd, has there been any uptick in genomic AI technologies coming through your pipeline and seeking this sort of certification?
Yeah, I would say so. There are lots of companies working in the pharmacogenomics space, so trying to predict, efficacy of drugs based on genomic profiles, and I think that's becoming a larger space that we're seeing. And then the other big space is using genomic data to predict treatments for diseases like cancer. And I think those are probably the two main buckets where we're seeing a lot of AI being used, and software IBDs being developed in the genomic space. Flick, is there anything else?
I was just going to say, I think there's sort of like a natural progression of the AI space in general and healthcare. Like we were previously saw a lot of radiology, but I would say actually the radiology, the amount of radiology that we're seeing is actually less, and it's being, a lot more varied, I would say, across not just genomics, but things like mental health and like pathology and things like that. So it's actually a lot more varied in terms of specialism, I would say, just in general, from a more high level perspective.
Brilliant. Thank you so much to both our speakers today. We've had a very informative, illuminating topic on the regulation of AI and genomics. Please join us again next month when we'll be having a new speaker. The proposed talk will be from a team who are using AI to write patient leaflets for ultra-rare diseases where there is currently no resource for the patient-facing population in genomic medicine services. I think it'll be a really interesting one to catch up. So thank you to our speakers, and thank you to our attendees for coming, and we'll see you again soon for the Genomic AI Network and Alan Turing collaboration on these webinars. Goodbye.
Thank you all.
Thanks, everyone.